Good afternoon. Thank you for joining us here. It's not every day that you get to interview a Time Magazine Man of the Year, a distinguished alum of Caltech, a generous interlocutor with the Caltech community, and a former trustee. David Ho is professor of microbiology and immunology at Columbia University, as well as a chaired professor of medicine, bringing together those two halves of the endeavor, and the founding director of the Aaron Diamond AIDS Research Center. He is widely recognized as one of the world's most influential physician scientists, thanks to his groundbreaking research in HIV and COVID. And we have the pleasure of exploring not only David's research and reactions to the various environments for medical research, but also to talk a little bit about how you got to this point. And so we'll talk for about 40 or 45 minutes and then turn over the floor to those of you in the audience who would like to ask further questions of David. So David, you immigrated to the US from Taiwan when you were 12 years old. What brought you to LA, and what was that transition like?
Before I answer that question, let me just say thank you, Tom, for the invitation to participate in this event. It's always a bit intimidating speaking in front of Caltech audience. In particular, I see David Baltimore sitting there. That's right. Who is obviously a hero to many of us. And he was chair of our scientific advisory board for a long period of time. And I'm grateful for his input. So what brought me to the US was my father had left Taiwan to come to the US to study in 1957. And he ultimately pursued an electrical engineering degree at USC. And he did not settle down and got a job until 1965. So for 8 years, we were separated. And he did not have the opportunity to return even a single time. So by 65, for those of you who follow recent American history, that was the full repeal of the Chinese Exclusion Act. LBJ signed that at the Statue of Liberty. And so we were, my mother, my brother, and I were among the first group allowed in. So I came as a 12-year-old, not knowing a single word of English, not even ABC. So it was a rough transition. The whole world changed for me suddenly. Instead of bicycle, we had cars. Instead of just the local market, we had supermarkets. So things were drastically different. But not knowing the language was the biggest challenge. And in some ways, my world was turned upside down, you could say, but it was a real new start and real opportunity. And I now attribute everything I've achieved to the event that my father actually went through and accomplished. But during that period, I went from sort of a good student to a dummy in the class, and also from, you could say, an extrovert to an introvert as a consequence of that. It took me many years to open up a bit.
And the number of Chinese students in your class must have been very small because of the change in the law. Is that?
Well, there were none other in my class, and in the whole school, it was just me and my brother.
I see.
But let me give you the real facts. In the first few months, we came in March, so it was toward the end of the school year, and my father was still living near USC, so we're in the middle of Watts. So we were the only non-black students for the first few months until we moved a bit north to the Griffith Park area the observatory area.
Right.
And then that was a much more diverse student body.
So did that influence the fact that your work has really addressed marginalized communities, often in the front in terms of AIDS, and very diverse parts of society? Has that been influenced, do you think?
It obviously had been a factor, but there are many other factors at play there.
Yeah. So is Taiwan still part of your life?
Indeed, it is. I return probably on average a couple of times a year. I'm engaged with a number of universities, National Taiwan University and others, as well as what is called Academia Sinica, which is the government research body.
Right.
And I'm a member of Academia Sinica as well. So I return. And Taiwan has evolved a great deal, and it's gratifying to see how it's developed over the years. Of course, there's always a constant threat that Taiwan faces.
Yeah. So even with the difficulties of coming here without English, you graduated at the top of your class at John Marshall High School. Was it obvious that you would come to Caltech? I mean, was that an ambition you harbored from the start or along the way?
Well, I would have to say that as an elementary school student in Taiwan, there were two heroes who were greatly worshipped, and they were both scientists. And it's Yang and Li in physics.
Sure.
I'm sure you know them well.
Oh, yes.
And so I just remember that they were glorified as national heroes. And so there was a pull from that. And then John Marshall High is fairly proximal to Caltech, and having an uncle who was an electrical engineering PhD from Caltech helped. And I must say that the community that we associated with were mostly Taiwanese scholars. And somehow that, I think, pushed me toward science and engineering.
And what made the biggest impression on you at Caltech, now entering here as a student?
I think just initially the intimidation that I feel right now, that everybody else is smarter, and the coursework was heavy. But what ultimately influenced me the most, I would say, is the critical scientific thinking, problem solving, not having to, I think it's still this way today, but not having to memorize a whole deal of things, and just taking problems, taking tests, and going back to your home or room and solving those problems, I think really influenced my subsequent scientific career, and in particular the quantitative emphasis of Caltech. And as Galileo said, the language of science is written in mathematics, right? So, and reflecting back, if someone asked me what is your major contribution to HIV/AIDS research, I would say it has to be the unraveling of the viral dynamics of how the virus replicates, and how that then inform treatment strategies. And all that actually requires quantitative thinking. And the Caltech, 2 years of mandatory college level math was very helpful, even though I think nowadays a high school student with AP calculus would be able to solve some of those issues. But it was that kind of thinking that allowed us to understand the relentless level of virus replication at tremendous rates, leading to tremendous mutational rates, which simply told us that confronting the virus with one drug at a time is doomed to fail. And yet, if you corner the virus by forcing it to make multiple mutations simultaneously in one single genetic background, the probability is each of those mutations multiplied. And knowing that, we went on to launch 3 clinical trials simultaneously in the mid-1990s to see if our theory was correct. And that's the beginning of the combination antiretroviral therapy era, and a strategy that's still employed today. And it was amazing to witness the success of that treatment strategy during that period. But subsequently, it was even more gratifying that that strategy was then applied to 20+ million people worldwide, particularly in sub-Saharan Africa, through Global Fund and through the President's Emergency Program for AIDS Relief. All of that nowadays is threatened. So, let's hope that's temporary.
So you talked about the quantitative aspects of understanding the replication. Were you able to model as well how to combine the various therapies, or is that much more a clinical result that you try?
Well, you have to take certain clinical factors into consideration. You don't want to combine drugs for medical reasons that should not be combined. Right. But you also want to know the mutational pattern of each drug and use that to inform how to combine the various compounds. And so that's how we addressed it. The other influence of Caltech on me, more on the sort of administration of science side, is the fact that, at a fairly young age, I was given the opportunity of starting this institute called the Helen AIDS Research Center, for which David served as SAB chair for some period of time. We wanted to sort of have the Caltech model, be small but mighty. And in fact, as it turned out, with a very small institute of less than 100 people, collectively, the institute was able to make a number of very important contributions in similar ways that Caltech has done on many diverse fields on a much larger scale.
Oh, it's certainly, as you point out, the secret of our success, and actually in terms of attacking big problems with really good people and having good communication and interdisciplinary approaches, which I know you share in terms of approach. The scalability issue is harder, but as you point out. So we turn back to the early days of AIDS. You saw some of the earliest cases here in LA. Can you describe what that was like at the time?
Yes, so by this point, I finished medical school, trained in internal medicine on the west side of Los Angeles. Yeah. And I had agreed with my chairman of medicine to serve an additional year as what is called chief resident. And it was during that period, around late 1980, early 1981, when a young man came into the hospital with a multitude of infections. And these are not everyday infections. These were what we call pneumocystis and toxoplasma. They're infections that would not plague a person with an intact immune system. And by then, we've seen lots of these cases in immunodeficiency patients who are on chemotherapy or transplantation medicines. And yet, this patient did not have any such history, transplant or chemotherapy. And previously, he was completely healthy. And so it was a medical mystery. We didn't know what was going on. His opportunistic infections were treated. He went home, but then died within a few weeks' time. And then surprisingly, another case showed up. And in rapid succession, 5 such cases showed up. And it mystified all of us, including the senior physicians, and we recognized that no such description existed in textbooks. So it had to be something new. And yet, it seemed like an immunodeficiency that was acquired and not inherited. There are a bunch of inherited immunodeficiencies, but this person had lived 30-some years without immunodeficiency, so it's acquired. So it's a medical mystery and a bit scary because all these 5 cases died within weeks after discharge. The common denominator was that they were all gay men, and they formed the initial report to the CDC. And subsequently, in mid-1981, CDC published what's called MMWR, describing these 5 cases. And a few weeks after that, a series of cases were described from San Francisco and New York. And these formed the initial description of what we now call AIDS. Then their blood samples were sent to UCLA's immunology lab, which had just begun to develop these markers for certain T cells. And Michael Godley ran that clinical immunology lab, and he found out that all of them had another common denominator, and that was the loss of what we call CD4 T cells, a very important orchestrator of the immune response. And that important cell was severely depleted, and hence explains the immunodeficiency.
Mm-hmm. Mm-hmm. Wow, it's a real detective story layered on with human tragedy. So.
Yeah, I mean, the tragedy is not just, succumbing to a deadly disease, but be shunned by friends and family and so on, and be devastated by the opportunistic infection. Some of them became blind. Some of them, had unrelenting diarrheal problems. I mean, and also very severe dementia for late-stage patients. So it's just tragic to witness all that. And I had gone into infectious diseases thinking that, for many decades, we were able to treat many infections. And yet, we were hit with a new disease that was essentially untreatable for the first 15 years of the pandemic.
Yeah. So you were there also at the early days of COVID-19 and did work tracking its spread. How is it evolving now? What is your prognosis of where we're going there?
Well, let me take you back a little bit.
Sure.
Aside from HIV AIDS, you might recall that 2002, 2003, there was a SARS outbreak, particularly affecting Asia, right? So it started in southern China in the Guangdong province, severely impacted Hong Kong and then rest of China and much of Asia. And by then I was somehow regarded as a virology expert, so I served as the consultant for the Chinese government, as well as the local governments in Hong Kong and Taiwan. So in the course of about 9-10 months, I visited those places for maybe 6 or 7 times. And I witnessed what was happening in Beijing and in Hong Kong. I mean, the streets were empty, the shops were all closed. And that was due to a coronavirus causing severe pneumonia that was transmissible from person to person. But that one was quickly brought under control with 800 deaths. So not much compared to COVID. Right. And yet it devastated those societies for a good year and a half. So I appreciated the impact, and when late 2019 came, I recall being on vacation for Christmas, New Year holiday, and I started to hear from virologists in China, whom I know, talk about a SARS-like pneumonia occurring in the city of Wuhan. And naturally, I paid a lot of attention to that, thinking this could be the recurrence of SARS. And so I followed that very carefully, had a number of text messages with colleagues from China. And then, I think it was January 3rd or 4th, the Chinese scientists published the sequence. Right. And it was not SARS, but it was a highly related coronavirus. And so naturally, I got very interested, and having dealt with SARS, paid a lot of attention. And within days, there's a Professor Zhong, who's sort of the Tony Fauci equivalent for China, came out and said this is due to human transmission. And within a couple of weeks of that, the city of Wuhan shut down. And having dealt with SARS, I knew this was good. You know, by the time, just think of SARS in Hong Kong in 2002, late 2002. Within days of recognizing a case, the virus was in 8 different countries, spread from a single hotel, the Metropole Hotel. Wow. And knowing that it took many weeks for Wuhan to shut down, we know the virus is out of control already. And so I think we were among the earliest group to jump on it. We paused our HIV research and say we need to take this on because we're going to be faced with a huge epidemic. I didn't think it would necessarily pandemic at that stage, but a huge epidemic. And it turned out to be the right call because we were then engaged quite deeply into this new virus research, and turned out to be one of our most productive periods, scientifically speaking.
Right. Yeah, and obviously with the miracle of mRNA vaccines, it's changed the complexion, but you still have a lot of residue of this virus. And do you have a sense of where it's going?
We were among a few groups who have studied the virus, SARS-CoV-2, and its evolution from the beginning till present day. And as you know, it's evolved a great deal. Yes, the mRNA vaccine made a huge difference, and other forms of vaccine, not so much applied in this country, but elsewhere. But you know, the alphabet soup that you all been hearing about, the alpha, beta, gamma, delta variant, and then Omicron, and then the BA1, BA5, and XBB, and all those. I mean, it's impossible even for experts to keep track. But the major point is virus continues to evolve under the pressure of our immune system, particularly our antibodies. And it's amazing to watch evolution in real time. And in this case, it's evolution of the virus and how it just keeps changing to evade our responses. And that's why the vaccines need to be updated. And now it seems, at least for the past 3 years annually, some of the calls were incorrect. One of the so-called bivalent vaccine didn't do much, but the subsequent 2 vaccines were made correctly and induced the better responses. And now FDA is poised to make another decision next month. And the data we generate help to inform that process. And so now, you know, every 6 months, it seems, we have another major variance. And this, I mean, for those doing life science, there have been, you know, incremental changes made by the virus by accumulating mutation in a stepwise fashion. But we also have major shifts in the virus. So they could occur by what we call antigenic jumps, right? Saltatory events or recombination events that led to major, Omicron is the first major saltatory event. And now we see how far the virus has evolved from the original Wuhan strain. And importantly, it should be noted that the virus has evolved this much. And when you look at the original virus and its relatives in the bat population, it's pretty small. So this antigenic distance is rather remarkable. And it's driven by our immune system.
Right. So at the height of the pandemic, the NBA kept playing with the so-called NBA bubble, and you were involved in that design. Can you tell us a bit about how that came about and what the strategy was?
Yeah, well, for scientists, I've been involved with the NBA a fair amount.
I see. That's another story in itself.
And for scientists who played basketball for Caltech a long time ago, never having won a single game.
You know, now there are NIF endorsement possibilities, so we could follow up.
So it actually began under Commissioner David Stern when Magic Johnson had HIV/AIDS. Right. And so I was involved as a consultant and provided some input and provide personal care to Mr. Johnson. But regarding COVID, it's actually a classic case of crisis management. In that, and it came about fortuitously, in the sense that I live in New York, I work at Columbia, and a fellow Chinese American, Joe Tsai, owns the Brooklyn Nets. Oh. And Joe and I know each other for some time, and he replaced Jack Ma as chair of Alibaba. And Joe invited me to the Lunar New Year party in late January 2020. And of course, he invited many other NBA folks. And then at the reception, at the bar, I bumped into Adam Silver, who's the current commissioner. Right. And we started just talking. I've known Adam not that well, but we met a number of occasions. We talked for a good 15 minutes or so. And of course, he knew I am a virologist, and he asked me about what was happening in China. And by then, Wuhan shut down, and the NBA China office shut down. Mm-hmm. And he asked me about it, and I said, one indication is to look at what's happening in Chinatown, because the Chinese community in New York knew there was something big happening, because it was relatively empty. And I said to Adam, it's coming. And next day he called me and says, could you help us plan for its eventual arrival? And I agreed. He wanted to pay me. I said, no need, I'll do this gratis. And so we struck up a relationship, and almost in the beginning, in the first few weeks, we talked several times a week. And then New York City, at Columbia Presbyterian Hospital, we have our first case March 1. And NBA has its first case March 9th. Mm-hmm. For those of you who follow basketball, Rudy Gobert came down with the infection, and Donovan Mitchell, who's his teammate at the time, came down with the infection a day or two later. And then March 11, NBA shut down, World Health Organization declared a pandemic, and the country shut down 2 days later. Right. And of course, that means loss of billions in revenue for the NBA. And so immediately they started to think about, well, how can we resume? Is there a way? And the initial impression in the first month or so was, no, not likely, given the state of the country. But gradually, all of us were forming our own tiny bubbles, right? At home, with our friends, with our family. And the idea was, what can we form a large bubble? Mm-hmm. it's just a matter of logistics, very complicated logistics. And so that idea of forming the large bubble came about that way. The credit goes to MBA. I mean, the execution is the difficult part, but the idea of the large bubble was from me as the principal advisor and several others. Mm-hmm. And it turned out that Disney World had lots of empty hotels, right? So you could form a large bubble and had lots of, I didn't know this, had many basketball courts. So you could have many games go on at the same time. Of course, with no audience, right? With fake audience on the side. And so there's testing, quarantine before you enter, daily testing, masking, good hygiene, and all that. And what they've done is incredible. There's not a single transmission within the bubble. After many months of resumed play. And it's written up in a number of papers, and MBA actually got a public health award for that.
And did you hear from the players during that period and how they reacted to the...
Well, initially, the famous local stars said, I'm not playing with no audience, right? But I think it was more important that they were able to resume the season and for the NBA to recover much of the revenue. And actually many other, the Olympics followed the NBA model to a large extent. But for baseball and football, it was hard to do that bubble because you don't have the right facilities.
Right. Even Disney World doesn't have multiple baseball stadiums. That's right. Dare I ask that we move on to bird flu, avian influenza, and ask what your view is about how that's proceeding and the chances of a vaccine, assuming some reasonable funding from NIH?
Vaccine is not a very popular word nowadays with the
It's
administration.
okay here.
I would say, first of all, there have been 3 major pandemics due to influenza virus over the past century, right? The famous one everybody knows about, 1918, 1919. But in the 50s and 60s, there have been 2 other flu pandemics. But what we now face is one particular variant of influenza called H5N1. And in particular, the clade is known to the aficionados as clade 2344b. So H5N1 was discovered long ago, but the first H5N1 that's thought to be pathogenic, highly pathogenic form of H5N1 was discovered in 1996 in Guangdong province in southern China. Then a year after that, it hit Hong Kong with 18 human cases. A third of them died. And then it was scary for Hong Kong for a while, but that was brought under control by culling the poultry.
Right.
Then this virus actually spread via migratory birds north and east, and north and west toward Central Asia, Europe, and Africa for the subsequent 20 years or so. And there have been a total of few hundred infections with a mortality rate of 50%. But most of those were not human-to-human transmission. They were mostly from birds to humans. So it had high mortality rate. Then in 2021, this particular strain, the 2344b strain, evolved in Europe somewhere and hit Newfoundland in 2021. And year after that, it hit the US and infected many birds, and including mammals. And in the subsequent year, it just spread down the west coast of Central and South America and up the east coast of South America. And just wiping out lots of mammalian species, including sea lions and seals. But we found out last year that 2024, that it infected American cattle a great deal. There are now probably close to 1,000 herds of cattle in the US and in about 20 different states. And at one point during the height, a quarter of the milk supply you could actually detect influenza RNA. So it's severe. It's now better because of intervention and testing. But important to note, this virus is rampant among birds and among many mammals. And the fact that you could have an avian virus infect mammals is very important because it's one step toward infecting a human.
Right.
And so I just think it's a huge problem. It's another pandemic knocking on our door, but there isn't a lot of attention paid to it. And in fact, what's happening to CDC and NIH is extremely detrimental to our response. So it's of great concern. But what's going for us is the fact that mRNA vaccines against this virus are already in the works. And also, in the population, we have some degree of cross-immunity to even this strain, particularly for the older population. So in my lab, we tested my blood and many other colleagues, and the older ones actually do better because of all the exposure history we have had to influenza and to some of the vaccines.
Yeah.
So some of us may be partially protected, but for the very young, they may not be as well protected. And the vaccines are in the works. And some of the drugs that are applied to influenza still would work. So we're much better off than the start of COVID-19. But on the other hand, given the history of H5N1, the mortality rate is a bit scary.
So we've skirted the general political situation. I don't want to get directly into that, but
We don't want to get Caltech in trouble.
well, I'm not sure the specifics are going to be that useful, except we could complain to each other. But the more general question of what we as scientists can do to influence policy, do you have some thoughts on that?
Yeah, I mean, it's certainly sad to me, and I think sad to many in the audience, what is happening with lack of respect for science, and in fact, lack of respect for truth, right? And you know, to a large extent, pandemic made all these worse because of the polarization that caused and folks using various restrictions related to pandemic to bash science. And now we see science being hit very hard at universities, including perhaps the hardest on my current institution, Columbia, which is now rather devastated with so many grants terminated. So it's tragic. I think as scientists, we have no choice but to speak up more, to continue to tell the truth. In science, we seek the truth, and when we know what is right, what is wrong, we should speak up. And I don't know if we could continue to acquiesce to some of the actions and demands placed on scientific institutions nowadays.
Thank you. One final question then. What would you tell students going into science and medicine these days?
Probably what I tell my students and my postdocs and to some extent the younger colleagues. I feel as a scientist, you have to stay curious. I mean, the curious nature in me drove me into science, I think. The other thing is, I think you have to be passionate in your chosen field. If you're not passionate about it, you're not going to be fully dedicated and pursue it with the vigor that's required. I also think that, you have to have some degree of conviction. I mean, for me, I took on the problem of HIV/AIDS when it was relatively esoteric, never realizing it would turn into a global pandemic. And yet, I had the conviction that it was a new mystery that needed to be solved, and that would have medical consequence. So having conviction, and also, being willing to take some informed risk, because certainly my immediate advisor, mentor said, well, don't pursue an esoteric disease like that. But yet, I held to my own personal belief and took some informed risk. And I think a degree of informed risk is needed. Other things, probably relate more to my experience running a lab. You have, I see my team members getting results. They then take those results and do all sorts of things. And sometimes they do things they shouldn't do. And so what I say is, figure out the difference between could and should, right? In English, I see people using those two words almost interchangeably. Mm-hmm. But if I use my Chinese head, and could and should come out very differently in Chinese. 可以做，应该做. And so I asked my lab members to figure out what is the critical path, and that's the thing you do. You could do many things, but it has no consequence. It's off the critical path. It's not the right thing to do. The other thing is, learn to differentiate dogma from truth. Mm-hmm. And sometimes I find myself saying things that sounds like dogma rather than truth. So I want my students or postdoc to challenge me on that. And my own example is HIV is considered a lentivirus. Lenti in Latin means slow. Right. And yet, it lulled the field into thinking, this is a slow virus, it doesn't cause anything for 8-10 years. And yet the virus is replicating away at enormous rates, right? So the dogma for the first, I don't know how many years of HIV research was completely wrong. So differentiating dogma from truth is very important for every scientist, but particularly the younger ones. Mm-hmm. And lastly, again, coming from my own lab experience with colleagues is, I've had many great students, but at the beginning, some of them, sit there and plan beautiful experiments, but don't execute very well. And others, just do one experiment after another, generating an enormous amount of data without synthesis. So I just emphasize both vision and action. Yeah. You need to plan strategically, but you also need to execute. And I always use a famous Japanese proverb to get the point across to them, and that is, vision without action is a dream. Mm. But action without vision is a nightmare.
Oh, that's wonderful. But the courage it takes not only to challenge dogma, but to actually get in the lab and take data is remarkable. I see that with my own students as well. Some of whom will build very intricate devices and take forever, and others who will build something that will fall apart the next day, but it will have collected all the data that's necessary. So thank you so much for those observations and insights into your trajectory, and want to turn it over to questions from the audience. I think with a mic in the back, is that correct? Yeah. So if you have a question, if you could just raise your hand and we can bring the mic to you. Oh, we have 2 mics. Please.
Hi, thank you so much. I'm a physician scientist in training myself and incredibly inspired to hear your talk. I was curious what you think about the prospect for vaccines that induce sterilizing immunity, such as mucosal vaccines, and where the pitfalls or challenges might lie.
Yeah, I think just take the COVID vaccine as an example. They've been great in protecting against disease and reduced hospitalization, death by over 90%, at least with the initial vaccine against the initial strain. But the virus is still getting through. And now we realize the immune response is great systemically and in protecting against severe disease in the lung and other organs. But it doesn't necessarily completely protect in the upper airways. And studies have shown that you could now use, in addition or in place, mucosal vaccines, and in certain circumstances achieve complete block of infection, not just the disease.
Maybe David?
On your right.
When did you come to Caltech?
I graduated in '74.
Graduated in '74. So you were in the middle of the development of the revolution of molecular biology.
Absolutely.
Did the molecular biology going on at Caltech at that time have any influence on you?
Absolutely.
Yeah.
That wasn't part of Tom's question, but I'm glad you asked. I came as a physics major. And high school biology was a bore to me, to be honest. You know, taxonomy and all that. I didn't like it at all. And so I like chemistry and physics. And there were inspiring figures around here doing that. Well, now and then, right? And Feynman was around, Gell-Mann was around, all these inspiring figures were around. This was a very natural draw. So I came as a physics, intending to major in physics. And the first course I took was with Lee Hood and Bill Wood. And it was inspiring. And as you say, modern biology was coming along, and it was not just inspiring, it was fascinating. And I would have to say, it was more tangible than theoretical physics.
I won't take it personally.
And at least to my parents, with more obvious applications. And so...
So did that influence you to go to medical school?
Yes, but I went to medical school wanting to do medical research, ultimately. So I have, other than the training period and the initial few years of being a medical school faculty member, I didn't practice medicine all that much. But I have to say the medical training was very important to me, providing a different perspective to a given biological challenge, particularly in medicine. And so we come at the problem differently from, say, the PhD scientists. Just as one example, we saw a lot of patients who did not get infected by HIV, despite lots of sexual exposures. And, why the outlier? And it turned out those people lack the receptor, right? These rare Northern Europeans who do not have CCR5 gene, or do not have an intact CCR5 gene, they were largely protected. And if they're heterozygote with one normal, one abnormal gene, they were protected from severe disease, even though they could be infected. So we see certain clinical problems and then take it back to the lab. So a different perspective that I think is very complementary to those with a PhD type of background.
Hello, yeah, this is Chen. I'm a geophysicist at Caltech, postdoc. I have a question. So, what's your sort on the scale of 40 to 50, maybe 100 year scale? Do you think COVID completely changed the trend of the world? Or we are seeing the change right now?
Over such a long period of time?
Yeah, long time period.
What I think will happen with the COVID virus or SARS-CoV-2 is that, every 6 months, every year, the virus is evolving. And you could say it's evolving for multiple reasons. For example, it could evolve because it has better affinity for the receptor on our cells. But that's not so. In terms of receptor affinity, it's variable over a long period of time. But what is predictably increasing is evasion of our antibody responses. So if we now look at related coronaviruses that have been in human population for a long time, there are 4 of them, common cold coronaviruses. We see periodic surges, and I suspect we will see that with COVID. Now it's at a low, but a year or 2 from now, I think we will see another surge. When I say surge, I mean number of infections. I think we will largely be protected from very severe disease. But it may ultimately become the 5th common cold coronavirus. But if, I talked about the saltatory events, the jumps of the virus. The major jumps have come out of South Africa. Why? And Southern Africa. And that's because we think there are lots of HIV-infected individuals there who could replicate the virus, allow the virus to accumulate serially the mutations, and then it gets transmitted. And if that virus happened to have an advantage, it's going to be dominant. And that's how Omicron came about. And that's another, and recently, what we call JN1 lineage virus came about. So events like that could lead to another surge and perhaps even some degree of severe disease.
So we have time for one more question. Yeah. Now the young woman, I think, in front of you was first. Yeah.
Hi, my name's Aline Kujakian. I'm a freshman studying biology. I was wondering what experiences during your undergrad led you to med school after being initially interested in biology?
Could you say the question again?
What experiences in your undergrad led you to med school specifically after being initially interested in biology?
I think it's related to what Dr. Baltimore had asked. I think just the exciting biology that was happening and its obvious implication to human health ultimately. So I thought I could have gone and pursue a PhD in life science, but I had certain had medical diseases in mind, and I thought that going to med school would sort of allow me to pursue medical research just as well as going to life science graduate program.
Great. So we have dueling microphones. Do you have a quick question?
I have a question, kind of on a tangent on her question. So I'm going to medical school next year, and I was wondering how you decided virology was going to be the topic that you wanted to research, and how of all the different topics you have in medical research that you wanted to make your contributions in this field.
Yeah, I didn't decide on specific area until much later. I think by the time I applied to medical school, I knew medicine and life science was my interest. And as I went through medical school, and you'll find this out yourself, as you do different rotations, you kind of develop interest in many different areas, and it's a constantly changing profile. But ultimately, I think after getting into medical training in internal medicine, infectious disease seemed like a very interesting area, not only for clinical practice, but for research. And then you have, inspiring figures in virology sitting in front of you. I think those things play a role. And David was doing his great work at MIT, and I was in training as a fellow, subsequent to medical training. And so a number of different factors went into that.
Well, thank you, David, for inspiring us. Really a great pleasure.